Genetic code. Today we are discussing the genetic code. Okay, genetic code. What is the meaning of the genetic code? Genetic code is a dictionary and corresponds with the sequence of the nucleotides and sequence of the amino acids. Genetic code is a set of rules by which is the information encoded in the genetic material like DNA and RNA sequences and translated into proteins by the living cells. The term is given by the George Gamow. Genetic code, the term is given by the George Gamow. Actually, to understand how proteins are the encoded and the begin after the structure of DNA discovered by the James Watson and Crick and Francis Crick. And George Gamow is the postulated that the three-letter code must be employed to the encode the 20 standard amino acids used by the living cells is to be build the proteins. Introduction to genetic code. The letters of the adenine, guanine, thymine, and cytosine correspond to the nucleotides found in DNA. They are the organized into the codon. The collection of codons is also called as the genetic codes. Collection of codons is also called as genetic code. For 20 amino acids, there should be 20 codons. Each codon should be have the three nucleotides to important specificity to each the of amino acids for a specific codon. Nucleotide, one nucleotide is the 4 combination. Two nucleotide is the 16 combination. Three nucleotide is the 64 combination. Is are the most of the suited for the 20 amino acids are they're given the 64 codons. Okay, genetic code 64 genetic codons. Next one, the genetic codons is also three bases. For example, ATG. Okay, genetic code is the expressed in simple table with the 65 codons. For example, okay, these are the genetic codon. Okay, genetic codes is the expressed for example, UCAG. Okay, UCAG. The triple E is the fill line is coded to the amino acid. Okay, then leucine, lysine, methionine, and AUG is the initiation codon is the methionine is the initiation codon. And next one is the UAA, UAG, and next one is the UGA. These are the three are the stop codons. Nonsense codons are the stop codons. Okay, actually, how many? 64 codons is there. In codon, it is this type of the genetic code dictionary consists of the genetic words called codons. Each codon consists of the three bases. Triple. There are the 64 codons. 61 codons code for the 20 amino acids found in the protein. Three codons do not code for the any amino acid. These are the nonsense codons. Okay. The types of the codons: sense codons, signal codons, start codons, and stop codons. What are the sense codons? The sense codons that code for the amino acids are also called as the sense codon. Only the code for the amino acids is also called as sense codon. These are the only they can code for the amino acids. This is the sense codons. Next one is the signal codon. These codons that code for signal during the protein synthesis is also called as the signal codon. What are the signal codon? For example, AUG. Okay, AUG, the methionine, UAA, UAG, UGA. These are the signal during the protein synthesis also called as signal codons. These are the AUG, some of the nonsense codons. Next one is the two types of the signal codons, terminating codon and initiating codon. Terminating codons also the nonsense codons UAA, UAG, UGA. These are the three are the termination codons for the nonsense codons for often referred to as the amber, opal, ochre. These are the terminating codons. Initiation codon. Actually, signal codons are the two types. Sense codons is the only this can code for the amino acids. Signal codons also, these are the code for the signal during the protein synthesis is also called as the signal codons. Okay. Initiation codons. AUG is the initiation codon. It code for the first amino acid first in all protein synthesis. Okay. AUG is the first initiation codon. These are the initiation for first amino acid. At the starting point, it codes for the methionine. AUG is also called as the methionine in eukaryotes for the formal methionine in prokaryotes. Age is the methionine. These are the initiation codon, especially in eukaryotes. Prokaryotes also the formal methionine is the initiation codon, is the starting codon. Anticodons, the base sequence of the tRNA which pairs the mRNA during the translation is also called as the anticodons. Anticodons are also, for example, tRNA, the oval position, they can see in the oval position codon 3 prime to 5 prime, 5 prime to 3 prime, mainly the mRNA, the tRNA also anticodon loop. Difference between the codon and anticodon. The codon could be present in the both DNA, RNA, but anticodons also is present in the in RNA. There is no in DNA. Okay, only these are the codons and anticodons. Codons and anticodons. Codons also only they are present in the DNA and RNA. Okay, codons could be present in the both DNA, RNA, but anticodons are always present in the RNA. Codons are written in the prime to 3 prime direction. Anticodons are as usually written to 3 prime to prime direction. Codons also mainly they are right to the 5 prime to 3 prime. These are the difference between the codons and anticodons. Codons also mainly they can see into the only in DNA and RNA. The both are the present. No, both are the containing the codons. Especially anticodons also they can see into the only RNA. These are the one difference, major difference between the codons and anticodon. The other one is the codons also is the right to return to 5 prime to 3 prime. And next one, anticodons also usually written to the 3 prime to 5 prime. Anticodons of some of the tRNA molecules have to pair with more than the one codon. Some of the tRNA molecules, anticodons also more than the one codon. Codons are the sequentially arranged in nucleic acid strand, while the anticodons also distinctly present in the cells with the amino acids attached or not. Codons defines Genetic codes. Major difference between the codons and anticodons. Next one is the codons defines the which is the anticodon should become next with amino acid to the create the protein strand. Anticodon helps the bringing a particular amino acid needs the proper positioning during the transcription. What are the characteristics of the genetic code? Triple code. These are the characteristics. Triple code, triple codon, comma-less. These are the characteristics of the all characters of the belongs to the genetic code. No non-overlapping code. The coding dictionary, degenerate code, universality of code, non-ambiguous code, chain initiation code, chain termination code. These are the all are the characters of the characteristics of the codons. Okay. Next one, what is the universality? The genetic code is the universal. AUG is codon for the methionine in mitochondria. The same codon AUG codes for the isoleucine in the cytoplasm. With the some exception noted the genetic code in universality. Okay, these are the universality. Non-ambiguous mean the genetic code is non-ambiguous. Thus, one codon cannot thus specify more than the one amino acid. The non-ambiguous code, non-overlapping is another characteristics of the genetic codon. One base cannot the participate in the formation of the mitochondria one codon. This means that is the codon is the non-overlapping. Continuous and the translation. The gene is the transcribed the translated continuously from a fixed starting point to a fixed stop point. Functions are not present between the codons. The code has the polarity. The code has the defined direction is the reading the methionine is which is referred to as the polarity. Reading of message from the left right and right to left will specify the different amino acids. For example, UUG is the stand for the leucine. From the right left, the GU is the stand for the valine. Okay. Degeneracy of the genetic code. Hobble hypothesis. What is the meaning of the Hobble hypothesis? Crick postulated the Hobble hypothesis is the account of the degeneracy of genetic code. According to this hypothesis, the first two bases of codon is the pair according to the normal base pairing rules with the last two base of the anticodon. Base pairing at the third position of the codon is the Hobble. Okay, Hobble hypothesis explain the degeneracy. Hubble hypothesis explains the degeneracy, the genetic code is the existence of the multiple codons for the single amino acid. Although there are the 61 codes for amino acids, the number of tRNA is the far less, which is due to the ovuling. Biological significance of the degeneracy of the genetic code. If the code were not the degenerate, 20 codons would designated the amino acids, and the 44 would be lead to the chain termination. The probability of the mutating up to chain the termination would therefore the much higher with the non-degeneracy code. Clinic significance. The mutation can be will explain the genetic code, point mutations, silent, missense, nonsense, missense and nonsense, frame shift mutations. The silent mutation, the single nucleotide change A to G, same amino acid is the incorporated. Mutation is unnoticed. Missense mutation, single nucleotide change A to C is the different anticodon amino acids incorporated. Loss of the functional capital protein. Nonsense mutation, Single nucleotide change from the C to T and stop is generated the perm- premature termination of chain may be the incompatibility with life. Frameshift mutation, insertion or removal of the bases can alert the reach the frame with the resultant incorporation of the different amino acids. Okay, these are the codons and anticodons. Mainly, these are they can use for the protein synthesis. Okay, protein synthesis, the process of the translation mechanism of the, already yesterday we discussed the uh, central dogma, the central dogma, DNA to RNA, RNA to the protein, DNA to RNA formation is also called the transcription, and next one RNA to protein synthesis they can form. These are also called as the is also called as translation. These are the central dogma. Especially the special codons, initiation codons are the start codon is the first codon is the messenger RNA transcript the translated by the ribosome. The triplets AUG and GUG play the double role in Escherichia coli. When they are occur between the two ends of the cistron, they code for the amino acids methionine and valine, respectively in the intermediate position in the protein molecule. But when they occur immediately after the terminator codons, they act as the chain initiation signals or starter codons for the synthesis of the polypeptide chain. Chain termination codon are the three triplets UAA, UAG, UGA do not code for the any amino acid and are also called as the stop codons. The three stop codons have been the named Amber UAG, Opal or Amber UGA, Okri UAA, Amber AMBER, Amber, Amber UGA, Opal or Amber UGA, UAG, UGA, UA is the Okri. They are also called as the chain termination codons because the codons are used by the cell to signal the natural end of the translation of particular peptide chains. The inclusion of any mRNA results in the abrupt termination of the message at the point of their location, even though the Mendelian inheritance. Next topic is the Mendelian inheritance. Mendelian inheritance is father of the genetics is the Gregor John Mendel, Austrian monk, born in 1822 near the Braun in the Australia. Is in pure family. The published his theory is the 1866 experiments on plant hybrids. In 1900, the work of the Mendel was the independently rediscovered by the Hugo de Vries, Karl Korens, Germany, Erich Tesmark, Austria. Mainly these are the work on the white flower and the purple flower. In F1 generation, they can form to the purple flower. Okay, is choose the why to choose the P plants. P is the easy to grow. They reproduce quickly. Emasculation and the pollination quite easy to the capable of the self fertilization. And statistical analysis in Mendel experimental is the mathematical probability, experimental prediction, testing experiment. Mainly they choose the seven characters: seed shape, seed color, pod shape, pod color, flower location, plant size. And seed shape is the round and wrinkled. And yellow is the seed color, yellow and the green color. And next one, pod shape, implanted and constructed. Next one is the pod color, green and yellow. Flower color, purple and white. Flower location, axial and the terminal position. Okay. And plant size, tall and short. These are the seven characters are they can choose the Mendelian. Mendelian laws of inheritance. These are the three types. Mainly law of dominancy, law of segregation, law of independent assessment. Some the gene. What is the mean of the gene? Gene mean units of inheritance usually occurring the specific location or locate on chromosome. Physically, gene is the sequence of DNA basis. There is a specify the order of amino acids in protein. Genes are the responsible for the heredity traits in plant and the animals. Allele, an alternative form a gene that occurs in the same locus in the homologous chromosomes. Homologous chromosomes. Okay, next one, allele, the alternate gene for the occurs in the homologous chromosome, allele, gene and allele. See, these are the okay, dominant allele and recessive allele. An allele that marks the presence of the recessive allele in the phenotype. Dominant alleles for the trait are usually expressed if any individual homozygous dominant or heterozygous. Recessive alleles, an allele that is the masked in the phenotype by the presence of dominant allele. Recessive alleles are the expressed in the phenotype when the genotype homozygous recessive. The uppercase letters are used to denote the dominant alleles, whereas the lowercase letters also denote the recessive alleles. Homozygous, what is the meaning of the homozygous? Having the same allele at the same locus on the pair homologous chromosome. Homozygous, homozygous also refers to the genotype consisting of two identical alleles of gene for the particular trait. Individuals for the homozygous for the trait are the referred to homozygotes. Heterozygotes, a genotype consisting of the two different alleles, a gene for the particular trait. Individually who are the heterozygous for the trait are the referred to the heterozygous. Next one, genotype. Genotype refers to individuals of the genetic potential, what kind of the genes and he carries, especially carries. Phenotype is the pheno meaning the show, pheno mean the outer. Okay, genetic is internal part. Refers to trait in the individual actually shows the outer structure. Okay, for example, homozygous mean capital P, small p, small p. Heterozygous mean, for example, Capital P, small p. These are heterozygous condition. Hybrid. Hybrid is the offspring that are the result for the mating between the two genetically different kinds of the parents. In F1 generation, the first offspring generation, the next and the subsequent generations are referred to as a F2, F3 monohybrid. Cross between the parents in differing in only one trait or in which only one trait is being the condensed. The hybrid cross is the cross between the parents in which two pairs of the contrasting characters are studied the simultaneous for the inheritance pattern. Next, first one is the first law is the law of dominancy. In cross parents that are the pure contrasting traits, only one from the trait will appear in the next generation. All offsprings are be hybrid for the trait and will have only the dominant trait express the phenotype. The phenotype trait that is not expressed the hybrid is also called as the recessive. For example, parental capital P, next one capital P is the purple flower and the white flower. In F1 generation also they can form the most of the purple flowers. Law of dominancy. And genotypic ratio is the law of dominancy, the 1 is to 2 is to 1. The phenotype ratio is the 3 is to 1. A law of segregation. Mendel's law of segregation states that the two alleles of genes that are the found on a chromosome pairs separate, with the offspring receive one from the mother and the one from the father. According to the Mendel laws, the two alleles act as the segregated fashion and do not mix with the or change each other. Mendel's second law, the law of segregation, states that these two alleles will be separated from each other during the meiosis. Specifically, in the second of the two cell divisions of the meiosis, the two copies each chromosomes will be separated from each other, causing the two distinct alleles located on this chromosome to segregate from the one another. This law of also referred to as law of purity of gametes, law of segregation, separation, law of segregation. Okay, law of segregation, especially true breeding parents, purple flowers cross with the white flowers. In F1 generation, all plants have the purple flowers. Okay, in fertilization among the F1 plants, in F2 generation, they can form to the 3 by 4th of the plants have the purple flowers, 1 by 4th of the flowers and having the white flowers. Okay, the law of phenotypic ratio is the 3 is to 1 in F2 generation. The next one is the genotypic ratio 1 is to 2 is to 1. A law of independent assessment. It states that the inheritance one character is always independent of the inheritance of other characters within the same individual. Law of independent assessment is based on the dihybrid cross. The alleles of the two more genes get stored into the gametes independent of each other. The allele received for gene does not influence in the allele received for another genes. The genes linked on a chromosome can rearrange themselves through the processing of the crossing over. Therefore, each gene inherited independently. What are the importance of the biological significance of Mendel laws? It showed the pattern inheritance, the characteristics of the parents, the offsprings. The law of independent assessment has laid down the foundation of inheritance, which claimed that the different genes associated themselves independently of one another during the gamete formation of the established the relationship between the dominant and recessive gene type. It also provided the reasons of variation offspring transferred from the parents to offspring in the form of the gene are present in the chromosome gamete. Next one is the post-Mendelian discoveries. Next one, the phenomena where the none of the two alleles in complete dominance is related to the phenomena where the none of the two alleles of gene dominant over the other, so that the when botons are present together, the new phenotype is found, which is the somewhat intermediate between the independent express of the two alleles in complete dominancy. Mainly examples of the incomplete dominancy mean they can cross with the red flower, white flower. In F1 generation, they can form the pink flower. Okay, pink flowers. Okay, the in F1 generation is the phenotypic ratio is the 1 is to 2 is to 1, and genotype is the especially in phenotype the F2 is the 1 is to 2 is to 1. Mainly these are the examples. Incomplete dominance show the Mirabilis jalapa 4 o'clock plant. Pure red and the white flowered parent produce the intermediate pink flowers. F1 cross, it gives the pure red white parents types along with the intermediate types of the intermediate types in ratio of the one red, two pink, one white. Okay, they have the pure forms of the black and white to be two are the crossed in F1 progeny is the blue color individuals in F1 generation. These are the 1 is to 2 is to 1. Codominancy, the phenomena which the both alleles of gene expression themselves simultaneously in heterozygote. Both alleles are the neither show the dominancy, recessive relationship nor incomplete dominance, but they express their the traits independently and they're known as the codominant alleles. In cattle, codominancy ABO blood group. Mainly in women's, ABO grouping contains the four blood groups types A, B, ABO. They are the governed by the gene, and it's the three alleles. Uh, IA, IB, IO, IA, IB. They are the dominant is of IO, but is the codominant with the each other. The produce the AB group phenotype. Multiple allelism. The existence more than two alternative forms of gene in a population occupying the same locus on the chromosome, it's homologous. It's known as the multiple allelism. Multiple alleles are the produced due to the repeated mutations of the same gene different directions. Human ABO blood group is the multiple allelism. Okay, pleiotropy, the condition which is our one genotype influence the more than the one trait, such the gene also called the pleiotropy. In a garden piece, example, the gene which control flowers colors also control the color of seed coat in the presence of the red spots in the leaf axils. In humans, sickle cell anemia and the phenylketonuria are the examples of the pleiotropy. Phenylketonuria serves as the prime example of the pleiotropy. It arises from the mutations in the phenylalanine hydroxide (PHA) gene impacting the breakdown of the phenylalanine to tyrosine in the liver. The pleiotropy effects includes the accumulation of the phenylalanine leading to the intellectual disabilities, changes in skin and hair pigmentation, and the neurological issues. These highlights are the signal gene mutations can be influence the various segments in the transfer. Next one is the polygenic inertancy. This type of the inertancy controlled by the one or more genes which is the dominant alleles is the cumulative effect with the each dominant alleles in the expressing part of the traits being shown the only when all dominant alleles are the present. The genes involved in such a inertancy are also called as the polygenies, skin color and the pigmentation. Always epistatis, the phenomena masking of the suppressing of the expression of the gene, the another allelic gene. The suppressor gene is known as the epistatic gene, and the suppressor gene is known as the hypostatic gene. Types of the epistatis dominant 12 is to 3 is to 1 ratio. In this, epistatis dominant for the own allele. Therefore, the effective on the heterozygous condition, example fruit color and the cucurbita pepper. Recessive in this type of the 9 is to 3 is to 4 ratio. In epistatic gene is the recessive to the own allele. Thus, epistatic gene can be only exact in homozygous condition. Example, Dominant epistatis, fruit color in the summer squash. Recessive epistatis, coat color in maize. Mendel experiment monohybrid cross, and next one is the dihybrid cross. Monohybrid in this experiment, Mendel took the two pea plants of the opposite traits, one short and one tall, and crossed them. He found in the first generation offsprings were all tall and called it is the F1 progeny. Then he crossed the F1 generation and obtained the both tall and the short plants in the ratio 3:1 ratio. The monohybrid, and the Mendel even conducted this experiment when the other contrasting traits like the green peas as versus yellow peas, round and wrinkled. In all of these cases, he found that the results were similar to the formulated the loss of the segregation and dominancy. The hybrid class. In the hybrid class experiment, Mendel considered two traits, each having the two alleles. He crossed the wrinkle and green seed and round yellow seeds. Observed that all the first generation progeny, F1 progeny, were round yellow. This meant that the dominant traits were the round shape and the yellow color. He then the self-pollinated the F1 generation obtained the four different traits: round yellow, round green, wrinkled yellow, and wrinkled green seeds. The ratio is the 9 is to 3 is to 3 is to 1. Okay. After the conducting research in the other traits, the results were found to be similar. From this experiment, Mendel formulated the second law of the inertancy, law of independent adjustment. Mainly conclusion of the from the Mendel experiment, the genetic makeup of the plant is known as the genotype. On the contrary, the physical appearance of the plants is known as the phenotype. The genes are the transferred from parents to offspring pairs known as the as alleles. During the gametogenesis, when the chromosomes are halved, there is a 50% of the chance one of the two alleles to fuse with the allele of the gamete. Mendel inheritance in human genetics. Many human traits, dominant traits, widow speak in the Huntington's disease, recessive traits, albinism. Next one is the recessive traits, albinism and cystic fibrosis, X-linked traits, hemophilia, Duchess muscular dystrophy. Okay, these are the Mendel inheritance. Text class. What is the meaning of the text class? Text class is usually is determining the homozygosity or heterozygosity. Dominant phenotype can be equally in the valuable in the determine the genotypes. Test cross. Test and back cross both are the breeding techniques used in the genetics and study of the inheritance patterns and determining the genotype. Test cross. A test cross is a genetic cross between the individual with an the unknown genotype and a homozygous individual. It helps the determining the whether the unknown genotype, homozygous genotype, and heterozygous. To identify the what are the purpose of the test class? To identify the genotype of an organism that shows the dominant trait. To confirm the Mendel laws of the segregation. In test class, for example, cross the consider the pea plants with the purple flowers, the cross with the uh, capital P, and small p. In genotype, capital P offsprings will the purple flowers, the capital P small p. If the unknown genotype, capital P small p, the offsprings will be the 50% of the PP and the 50% white. In test cross, in monohybrid cross ratio is the 1 is to 1 ratio, and dihybrid test cross ratio is the 1 is to 1, 1 is to 1, 1 is to 1. Mainly, test cross dihybrid genetics is also okay. Next one is the back cross. Back cross is a genetic cross between the offsprings F1 and one of the parents or individual with the similar genotype. What are the purpose of the back cross? The preserve the describe the traits in the breeding to study the inheritance pattern by the reinforcing and parental traits. Types of the back cross, dominant back cross, and cross was the F1 generation, no homozygous dominant parent, the offspring 100% dominant phenotype, recessive back cross. In F1 generation, homozygous recessive parent, small p, offspring the 50% dominant and 50% recessive. This type of the back cross also uh, main, mainly in test cross also uh, in F1 generation gamete they can fuse with the recessive characters. Okay, they can fuse with the recessive, they can form the test cross. What are the difference between the test cross and back cross? And test cross is the cross of an individual with the unknown genotype with the homozygous recessive individual. In back cross, cross with the F1 offspring with the one of the parents. Okay, mainly the cross between the unknown parents. Test cross and back cross. Next one, modification of the 3 is to 1 phenotype ratios, when incomplete dominance, codominance, multiple alleles. Okay, next one is the linkage. Linkage is the tendency of genes located close together on the same chromosomes to be inherited together during the meiosis. This concept was the first discovered in the Thomas H. Morgan while studying of the fruit flies, Drosophila melanogaster. Linkage is the contradicts the Mendel laws of the independent assessment, which state the gene different traits assessed independently. Types of the linkages: complete linkage occurs when the genes are very closely together on the same chromosome and are always inherited together. No crossing over occurs between them. An example, Drosophila genes controlling the certain body color and the gap is the complete linkage. And incomplete linkage, the partial linkage, occurs when genes are the relatively close but can sometimes be separated due to the crossing over. Crossing over between the homologous chromosomes and during the meiosis can exchange alleles, producing the recombinant offsprings. Linkage groups. Linkage groups is a set of the genes located on the same chromosomes that tend to be inherited together. The number of linkage groups in the organism equal to haploid chromosome number. Humans have the 23 linkage groups. Factors affecting of the linkage group. Distance between the genes. The closer genes have the stronger linkage, and the distant genes are the more likely to be separated by the crossing over. Chromosome location is also a factor affecting of the linkage. Genes near the chromosomes hence the experience more crossing over than the genes near the center. Environmental factors, temperature, radiation, and chemicals are influence the crossing over linkage. Linkage and crossing over. Crossing over occurs during the prophase 1 of meiosis, where the homologous chromosome exchange of the genetic material. Recombinant frequency is to the measure the linkage. High high recombination frequency genes the further apart from the linkage. Low recombination frequency number of recombinant by total offsprings into 100. Linkage is the Morgan experiment in Drosophila, especially studied the body color. The expected the 9 is to 3 is to 1 ratio to independent assessment occurred, but the observed the high number of partial types of the due to the linkage. Linkage genes, human red genes, color blindness is the hemophilia, X-linked genes often inherited together. The sickle cell anemia and the beta thalassemia genes is located in the closer together chromosome level. What are the impedance of the linkage? Helps the gene mapping in determining the distance between the genes. Explain the expectation of the Mendelian inheritance. Useful in plants and the animal breeding to develop the desired traits. Helps the understand the genetic disorders and inheritance patterns. That the linkage, chromosome mapping. Chromosome mapping is the process of determining the relative positions of the genes on a chromosome and measuring the distance between them. It helps the understanding gene linkage, recombination, and the inheritance patterns. Types of the chromosome mapping: genetic mapping, linkage mapping, determines the relative locations of the gene-based recombination frequency during the meiosis. Measures in map units, centigrams. Okay, physical mapping determines the exact location of the genes and chromosome using the molecular techniques. Is the base pairs units to the measure the distances. The techniques fluorescent in situ hybridization fish and restriction mapping, DNA sequencing, cytogenetic mapping uses the staining. The techniques in identify the specific chromosomal regions where the regions are located. G bonding and karyotype. Principles of the genetic mapping. Genes that are the closer together have the low recombination frequency. Genes that are the far apart have the higher recombination frequency. Crossing over during the meiosis influence the gene distances. Class calculation of the genetic distance recombination frequency equal to number of recombinant offsprings by total offsprings into 100. Okay, importance of the gene chromosome mapping. Gene mapping identifies the location of disease causing genes, cystic fibrosis, hemophilia. Evolutionary studies helps the compare the genomes of different species. Breeding programs assess the plant and animal genetics and improve the traits. Human Genome Project (HGP) uses the advanced mapping to the sequence of the entire human genome. Examples of the chromosome gene mappings: the Huntington's disease gene HTT mapped to the chromosome. These are the gene mapping. Gene mapping. Sex linkage. The sex linkage is the genes located on the sex chromosome X and RY that are the inherited differently in males and females. The genes do not follow the typical Mendelian inheritance because the expression depends on the sex of the individual. Types of the sex linkages: X-linked inheritance, gene located on the X chromosome. Since males have the only one X chromosome, XY, they express the X-linked traits more frequently than the females, XX. Females can be carries heterozygotes, but the male cannot. They have the only one X. X-linked, X-linked, especially examples of X-linked disorders: hemophilia, blood clotting disorders, red-green color blindnesses, ability to distinguish red and the green, Duchenne muscular dystrophy, muscular wasting diseases. Example of the X-linked recessive inheritance mainly carried the mother. Y-linked inheritance genes located in the Y chromosome. Only males inherit the Y-linked traits. Since females do not have the Y chromosomes, traits are the passed directly from the father to son. Examples of SRY genes controls the male sex determination. Hairy ears syndromes found in the some of the population. Pseudomonal regions. The importance of the sex linkage explain the some disorders affect of the males more than the females. Helps the genetic conclusion and the counseling predicting inherited diseases. Important of the evolutionary biology, sex-linked genes can affect the natural selection. These are the X-linked sex linkage. Okay, mainly sex-linked inheritance in man's. Women-linked disease, the albinism, various forms, cleft plate, hemophilia B, Christmas diseases, hypoxanthine guanine, the phospholipase and one transferase deficiency, muscular dystrophy, night blindnesses, Parkinsonism, tyrosine binding globulin production. The criteria identify the sex-linked recessive traits. The pedigree studies is as the follows. The trait occurs the more frequently in males than the females. The traits are the transmitted from the affected man through the his daughters of the his grandsons. X-linked allele is the never transmitted directly from the father to son. Allele affected the females have the affected father and the carrier is the mother. Y-linked sex chromosome genes, the Hollandric genes, they are the occur normally Y chromosome, the gene that determines the excessive hair development of the on the ears. Hypotrichosis is the Hollandric histochemopathy antigen genes located in the on Y chromosomes in women. The mouse, rat, guinea pigs, they controlling the segmentation of the long arm of the Y chromosome. These are the X-linked chromosome. Okay, thank you.